Welcome. Hereby, I open this academic ceremony in which Nikita van der Burgt will defend the academic thesis, The Many Faces of Psychosis, a novel approach to identify and classify rare forms. May I invite you to present a summary of your study and the conclusions of your thesis.
Dear Prorector, dear members of the Corona, dear colleagues, dear family and friends, in the upcoming 50 minutes, I would like to share some of the highlights of the research that I've been conducting for the past 5 years, which are written down in my thesis entitled The Many Faces of Psychosis, a Novel Approach to Identify and Classify Rare Forms. Psychosis is a mental health condition in which individuals may see or hear things that others do not. Additionally, they may perceive things differently, which we call hallucinations and delusions. Additionally, individuals may experience memory problems and attention problems and no longer enjoy things that they used to, which we call cognitive and depressive symptoms. It is estimated that up to 3 out of 10 individuals of the general population will experience such symptoms once in their lifetime, which means that several individuals in this room may have experienced those symptoms in the past or will so in the future. Hence, the occurrence of these symptoms is much more common than what is often thought. However, only if these symptoms occur more often and when they affect an individual in their daily life, consistently, patients have a psychosis spectrum disorder. Current treatment often focuses on medication and psychological treatment, which focuses on reducing and dealing with the symptoms. Although several treatment options are available, not everybody responds similarly or as expected. This may be because underlying causes are diverse and often not fully known or understood. However, it is known that in some cases, psychosis can be caused by another disease, an underlying disease. My research focused on several of those possible other diseases, among them a genetic disease and an inflammatory disease. It's important that we identify these underlying diseases because they are treatable with either lifestyle changes or a different type of medication. Hence, the traditional treatment may not work for these patients. And not providing the appropriate treatment may result in permanent symptoms or permanent damage, and in some cases, even death. First, I would like to tell you a bit more about those genetic diseases. These genetic diseases consist of a group of genetic disorders, which are collectively referred to as inborn errors of metabolism, or short IEMs. IEMs are characterized by mistakes in the DNA of an individual, which are often inherited from the parents. These mistakes in DNA may result in problems with an individual's metabolism. For example, someone with an IEM may not be able to break down certain types of food. These disorders may appear early in life or later in life, namely during adolescence or adulthood. My research focused on adolescence and adulthood. In case the disorder appears later in life, mental symptoms such as psychosis, which I described previously, but also depression and anxiety are the most commonly observed. For example, individuals may hear and see things that others do not see or hear. Or they are anxious or do not enjoy things that they used to enjoy. It is also possible that they have trouble remembering things. And although these symptoms are not specific for the disorder, and they occur in many other disorders, the longer people suffer from these disorders, while untreated, more symptoms will occur. For example, the mental symptoms may be followed by more visible symptoms, such as epileptic seizures. However, psychosis may be the most prominent symptom early in the disease. Currently, when IEMs are diagnosed or are manifested during adolescence or adulthood, they are usually diagnosed after physical examination, several tests in the blood and urine, and often scans and ultrasounds are needed. However, a certain result in one of these tests may not lead to the diagnosis of an IEM. Therefore, the results of all these different tests have to be combined in order to diagnose an individual with an IEM. This results in a very time-consuming and expensive process. Additionally, it's considered a difficult and unpleasant process for the individual itself. Consequently, it may take a long time before patients receive an appropriate diagnosis and treatment. However, the earlier the treatment is started, the less likely an individual is left with long-lasting symptoms. Diagnosing patients as soon as possible is therefore very important. Consequently, we were wondering whether we can speed up this process. Is it possible to not only screen for those IEMs in patients with psychosis, but also to do this in a way more efficient way as what is currently done in the hospital? We believe so. A possible tool for this is next-generation sequencing, or short NGS. This tool is able to detect these mistakes in the DNA that I was talking earlier about. More specifically, NGS enables us to detect the mistakes in the DNA that are associated with a number of treatable IEMs. Hence, they are treated with lifestyle changes or supplementation of vitamins. For this test, you only need one blood tube, so no longer ultrasounds, scans, or additional testing. Therefore, it's not as unpleasant as the different tests that are currently used in the hospital. And that's why in Chapter 3 investigated whether this tool could be used to detect these mistakes in the blood of patients with psychosis. We compared our findings with the DNA of healthy individuals, which we call controls. In this study, we found no patient with psychosis or controls had an IEM, so no patients were affected. However, we did detect variants of those mistakes in the DNA that on itself do not cause the disease, but if they would be combined, and you would be very unfortunate, they maybe would be causing the disease. Consequently, depending on the combination of these variants, you may either be healthy or you might get sick. For these patients, it means even though they are not affected by an IEM themselves, they are a so-called carrier. They carry the disease or a variant for the disease. Hence, when planning a family, individuals may have a child with an IEM in case the partner carries a variant as well. So even though the patients themselves did not differ from the controls, knowing that you are a carrier is actually still relevant. Why? Because being a carrier for such mistakes in the DNA may be associated with more mild symptoms or other problems that you may be able to develop later on in life. However, whether that's actually the case is not yet known. Additionally, being a carrier may be of influence when establishing a family, as individuals who both are a carrier of a variant may have a child that's actually affected by an IEM. From these genetic diseases, I would like to shift the focus to an inflammatory disease, which we call autoimmune encephalitis. Autoimmune encephalitis is characterized by the presence of so-called autoantibodies. Antibodies are molecules produced by the immune system when a bacteria or virus enters the body. These antibodies help fighting against those intruders, which makes sure that you are healthy again. However, it's sometimes possible that these antibodies attack the body itself instead. In that case, these antibodies are called autoantibodies. In the case of autoimmune encephalitis, these autoantibodies specifically target the brain. This results in inflammation in the brain, which may lead to similar symptoms as I mentioned before, such as hearing things that other people do not hear, feeling anxious, depressed, or having difficulties with your memory. Again, psychosis is often the first and strongest symptom of the disease. Currently, autoimmune encephalitis is diagnosed after physical examination and testing the blood and preferably some fluid from the brain for the presence of these autoantibodies. These autoantibodies are detected by a specific technique in which we specifically created cells are used in a petri dish. With these methods, we can study the impact on the cells on our body. It's like finding clues in your blood, or brain fluid in this case, to understand what's going wrong in your body. In the case of detecting autoantibodies, the cells are mixed with a sample of the blood of the patient, or brain fluid, and if their autoantibodies are present, something happens to the cells. They change in color. By looking at the color of the cells, we can conclude whether the blood or brain fluid of a patient contains the autoantibodies. This is a method that is currently used in the hospital to diagnose patients with autoimmune encephalitis. However, this technique only allows us to detect 6 targets to which these autoantibodies can bind. Although they are the 6 most commonly observed targets in patients with autoimmune encephalitis, there are many more targets that these autoantibodies can bind to. Hence, autoimmune encephalitis can also be the result of autoantibodies that bind to targets that are not measured by the currently used technique in the hospital. In order to better identify patients with psychosis due to autoimmune encephalitis that may not be diagnosed with currently used methods in the hospital, we conducted 2 studies. One study focused on improving the identification of these patients by adding a diagnostic technique to the current technique, and the second study focused on a new target that is not measured in the hospital routinely. First, I would like to tell you a bit more about the first study that we conducted. In this first study, we tested the blood of 45 patients and the brain fluid of 1/3 of these patients. And we tested whether the blood and the brain fluid contained these antibodies with a different technique than what's currently used in the hospital. We also assessed the severity of their symptoms and their functioning in daily life. Furthermore, to detect possible neurological symptoms, which may be subtle in this case, the patients underwent a neurological examination as well. For this different technique, we do not use cells, what is done in the hospital, but we use rat brains to visualize the binding of these autoantibodies. With this technique, we are able to screen for a higher number of targets compared to the technique that's currently used in the hospital. Again, similar to the technique with the cells, a change in color indicates that the blood or brain fluid of a patient contains these autoantibodies. So in chapter 4, we use this technique to screen for the presence of autoantibodies in a total of 45 patients. We found that in one of those patients, autoantibodies seem to be present in its blood, out of the 45 patients in total. However, to which target these autoantibodies bind exactly is not yet known, but we need to know that in order to be confident that these autoantibodies play a role in the psychotic symptoms of this patient. Therefore, further research is still necessary. But besides the targets that are screened for by the use of those rat brains, there are still targets that we're not able to measure by the technique. Recently, a new target was described in patients with psychosis. However, this new target is not screened for in the hospital routinely, and we're not able to screen for that by the use of the rat brains as well. Hence, that's why we conducted this second study, in which we combined techniques in order to measure this new target. Hence, in chapter 5, we combined those different techniques. We combined the techniques with the cells, which is done in the hospital, but now specific for this new target. Additionally, instead of looking at the cells to see whether they change in color, we actually measured the number of antibodies that are present. We did this by measuring this new target in the blood of 262 patients with psychosis and 160 control individuals. Of all patients and controls that we tested, 4 patients and 1 of the controls tested positive for these autoantibodies. And although we still have to further research to confirm whether these autoantibodies are actually related to the symptoms that these patients experience, we did notice that 2 out of the 4 patients that tested positive for these autoantibodies experienced visual hallucinations, which means that these patients saw things that other individuals do not see. Additionally, out of the 4 patients, 1 of those experienced cognitive symptoms as well, so they may have trouble remembering things. Individuals, both are indicators for an underlying disease. Summarized, the fact that some patients with psychosis have an underlying disease is supported by the findings written in this thesis. And although these underlying diseases may be rare individually, together they are common. Doctors should think of a possible underlying disease, especially in case of specific symptoms, among them those visual hallucinations or severe cognitive symptoms. In that case, additional tests may be beneficial in diagnosing these patients. Furthermore, we propose specialized centers for each underlying disease, where patients can go to during the process of both diagnosis and treatment. For example, within these centers, specialized doctors are situated, which are familiar with those diseases. Additionally, within these centers, current and additional tests can be performed. And finally, patients can go there for the appropriate treatment. With that, patients may benefit from a better quality of life, and long-term symptoms may be avoided as possible. And with that message, I would like to thank you for your attention and give the word back to the prorector.
Thank you for your presentation. Before we start the opposition, I would a special welcome to the promotion team, Professor Dr. Therese van Amersfoort, Professor Dr. Pilar Martinez, and Dr. Nico Leibold. So now the opposition will be opened by Professor Bart Rutten, who is having a chair in psychiatry at Maastricht University and has a double function. He's also secretary of this session.
Thank you, Mr. Prorector. Dear candidate, I have to say it was a pleasure reading your thesis. Wonderful work, very inter-cross and multidisciplinary in nature, the work that you've done. I just want to compliment your promotional team. That was spread across different divisions in the research institute that we are having here in Maastricht. It is not that easy to, on a daily basis, walk into each other's offices. And you managed to do real translational work on a diverse range of biological entities and inborn areas of metabolism, autoantibodies, and the lipids. So that's amazing and very important fundamental work. And I would like to go over to one of my questions. So the question would be an opening question in the sense that you have been touching upon this a little bit during your presentation, calling them underlying diseases. But I was wondering, so you've been studying 3 biological entities, but you try to link them to psychosis. And I was wondering, do you envision that there are multiple pathways towards a certain phenotype, or are multiple factors contributing to a common cascade of events that ultimately leads to a certain clinical expression phenotype? How would you place your findings in a conceptual model of etiopathogenesis of psychosis?
Dear highly esteemed opponent, thank you for your kind words and your question. First, I would like to emphasize that although I focused on psychosis, for example, in the case of inborn error of metabolism, which you can also read in chapter 2, it's actually not limited to psychosis. Patients also experience a more variety of symptoms among the depression, anxiety, even intellectual disability is often present already during childhood, but what is often not diagnosed like early in life. But the same for autoimmune encephalitis, it's not limited to psychosis. These autoantibodies, depending on to which target they bind, actually, different symptoms may appear. So there's also research been done in, for example, depression and anxiety, which probably means that there's a common pathway involved. However, of course, we do know that for psychosis, the NMDA receptor is very important. And it kind of makes sense that in that case, these autoantibodies, often in psychosis, bind to the target of the NMDA receptor, because in that case, you often get psychosis as a symptom. However, if they bind to another target in the brain, you may see a manifestation of different symptoms. For example, also more motor symptoms, that's also possible. It all depends on which indeed pathway is affected. I'm not sure which pathway is affected. I think that still needs a lot more research. And I think also for the inborn errors of metabolism, we do know that the metabolism is affected due to a mistake in the DNA, but we do not know exactly how in the end this leads to this clinical manifestation, other that, for example, toxins cannot be excluded from the body or things build up. So I think it's a good idea for further research.
So it's a very broad field, and maybe you want to retitle your title in the many phases of mental illness rather than psychosis. Thank you very much. I was also wondering, so you referred to this in your introduction, I think. You talk about gene-environment interactions, and that they are probably also important in onset and course of mental illnesses. Now, that's all that I read about the environment in your thesis, I think. But I was wondering, how do you potentially link environmental factors to your work? I mean, in the field of autoimmunity, I can imagine this easily, but otherwise, do you have some ideas on this?
Yeah, in case of autoimmunity, indeed, well, previously it was thought that, for example, in autoimmune encephalitis, it was most commonly in young females, and they actually had a teratoma, which is a tumor, by the way. And actually there, the autoantibodies were produced, going to the brain and affecting the individual by causing psychosis. But we know now that actually only a specific part of those patients have a teratoma, and with that of a clear origin. So it's indeed true that for many psychotic spectrum disorders, it's thought that there's a gene-environment interaction, of course. So a certain mutation in the DNA makes you vulnerable for the disease, but it doesn't mean that you actually develop the disease. I think for these underlying diseases, especially the inborn errors of metabolism, you actually already have the mutation in that sense. It is possible that you have a mutation that you have because they are homozygous if you have an IEM, so you have two variants of the disease, but it's also possible that you have, they are called compound mutations, so you are heterozygous for one variant of the gene, and you're heterozygous for the other. I'm not sure what is needed in the environment for that to occur, but I do know, or I do believe actually in this case, the environment may play less influence compared to patients that have a psychotic spectrum disorder due to other reasons.
Okay, many thanks. Food for further investigations, I think.
Definitely.
So my third and I think last question would then refer to chapter 4, in which you investigate the prevalence of neuronal surface antibodies in the PSYCHOSIS study. And you found on page 115, you described one positive case, right?
Yes.
I was wondering, could you comment on what you think is happening there? Is this a false positive finding? Is this a true finding, now meaning something on causality? Or is it an epiphenomenon? Because you also thereafter described the case, and then I was also wondering as a clinician, okay, that's nice that you have some ideas, but what is then next? Has this been reported back to the clinician and the patient, and was there any clinical implications of the finding that you did? So multiple questions wrapped into one.
Yeah, I like this question actually. So it is indeed perfectly possible that this patient is false positive. However, as you may aware, we test the sample several times to make sure that what we see is actually visible multiple times and not just a once occurring finding. In order to actually diagnose a patient, you need to be sure that you need to identify the actual target. So for this method with the rat brains, we only screen for possible autoantibodies. However, we do not know the specific target yet, and that would be the next step, which we do by the cell-based assays, which still need to be done, but the project is approved, so that can be done very soon. And then if we know the target, so for example, the NMDA receptor or GABA-B or AMPA, in that case, we're going to do the live staining with the neurons to actually see if these autoantibodies bind to the surface of the neurons, because it's thought that if they bind to the surface of the neurons, they are actually pathogenic. So for this patient specifically, we were not able to draw CSF or the brain fluid, but we are actually working on getting another blood sample and test that as well to see indeed if we can again identify those antibodies, and if we then can identify them, and then I will definitely contact the clinician to ask for CSF, so we can test that, and then further on, of course, inform the clinician about that, and the clinician can then make a decision regarding treatment.
Thank you very much. So you would require replication. And before giving the word back to the provider, I would like to again thank you also for providing the opportunity to many clinical residents to visit the lab, and that you have been disseminating your knowledge to clinicians. So many thanks. I hereby give the word back to the provider.
The opposition will be continued by Professor Dr. Clara van Karnebeek, having a chair in personalized medicine of metabolic diseases at the Emma Center for Personalized Medicine at the Amsterdam University Medical Center. As the University of Maastricht, we consider it very important that also opponents from outside are present. So welcome. The floor is yours.
Thank you, Mr. Prorector. Dear candidates, I also would like to start with congratulating you, your supervisory team, and your team, friends, family, because this is a victorious day, as you state in one of your propositions. You've done your best, and this has resulted in a great body of research, which is both multidisciplinary and translational, but it evokes many questions for me. And so I would like to start out by asking your paranymph to read proposition number 6.
Personalized medicine is essential to improve the lives of patients.
So, as a pediatrician and geneticist, I would like to agree, but I don't really understand what you mean with personalized medicine. So, how will it improve the lives of patients who experienced psychosis?
Dear highly esteemed opponent, yes. Thank you for your kind words and your question. I believe, and that's why it is in the statement, that personalized medicine is very important, and I'm happy to tell you a bit more about it.
And what is it? Very short.
Yeah.
What are the elements of personalized medicine?
It would be that actually you treat the underlying disease or the cause of disease rather than only the symptoms.
I think that's a nice start, but very optimistic. So if you consider there are 1,800 and counting, previously called inborn errors of metabolism. We don't use the term anymore of an error, but let's go with the flow. Only less than 10% is actually treatable, although diagnosis, as you mentioned, would give opportunity to improve lives via genetic counseling and perhaps personalizing care. So I think the treatment is something which only for a subset of cases is reality. But I agree with you, we should diagnose these patients. And what you've done in chapter 3 is admirable, but it's from a while ago. Technology has moved on. And so what I would like to ask you is to take a look at table 2 on page 96. You discovered or identified rare variants in 6 patients. And what is the next step in diagnostics to evaluate whether these have an impact on protein function or whether these are the cause of disease?
Of course, it's very important that actually now we found those variants that we also translated back to the patients, of course. So for some of those variants, it is known to which inborn error they are related to. For some, not yet. Fun fact, several patients had the same actually variant, and we were called to by the lab in Amsterdam to confirm whether or not they were actually related, because it's indeed so rare that actually several individuals have the same mutation. I would say the next step, but it's really difficult because these are so rare disorders, it's really difficult to get funding to do further research on it. But ideally, what I would propose to actually combine chapter 2 and chapter 3 to use, for example, the screener that we proposed in chapter 2 within the clinical field. And if psychiatrists actually start working with that and we can test whether it's feasible to have that in practice, only the subset of patients indeed that have those atypical symptoms, we're able to pick them out of the general psychosis spectrum disorder.
To screen only
and to screen
those,
only
yes.
those patients also to limit costs, for example.
Exactly. Yes. I could go with that. I think for some of these genes, there are metabolic tests which can show whether there's an abnormal metabolic profile or not, which would either conform or exclude the disease. Because what's obvious is that also the controls carry many variants. Yes. And so what I'm wondering about, would it be worth looking for the second variant, which could be hiding in these patients, for example, in introns, or is not picked up by the gene panel, so pursuing it further, because I think patients sometimes become worried of being a carrier, and what does it mean? And so if I understand correctly from this table, of the 60 patients, no one was identified with an inborn error of metabolism. And yet you say we should go ahead and implement next generation sequencing. And I would agree with you, we have to move ahead, but we need more evidence. And so how would you design the ideal study for your successor to do? Let's start out maybe with, would you go for a gene panel of 57 genes? Or would you say, no, let's go broader, let's screen all 20,000 genes for variants, or somewhere in the middle?
That's a good question. So if money would not be a limiting factor, to be honest, I'm not sure. I know that it's now possible at the current date to screen for more treatable IAMs compared to when we published this paper. Ideally, maybe you would screen for all of them, although you're indeed like wondering what is the benefit in that case, because you don't want to have the patients to be worried if there's nothing to be worried about. And of course, it's also possible to have those variants in the controls. But my first step would indeed to repeat the study, do then the improved panel of treatable or variants of IAMs that are treatable, and then in a way larger cohort to be sure that it's indeed not a coincidental finding, but we can actually see it in many more patients.
Yes, because these are all very rare diseases,
Yes.
right?
So you need a large cohort.
Yes, I could agree with that. And so why screen only for the treatable diseases? Because is it not very important to know whether there's an underlying cause of disease and maybe opportunity for treatment in the future?
Yes. However, what we do see that mainly the IAMs that are manifesting in adolescence and adulthood are usually the treatable ones. So the more severe types of IAMs actually manifest early in life. So of course we have the newborn screening in which some of them are screened for, but we actually miss a lot of them. Ideally, you would actually screen for the ones that are not screened for during the newborn phase.
Newborn screening, yes. So I think with next generation sequencing, we are actually identifying many very mild forms of these classic metabolic diseases which presented during early childhood. And I think also mild forms can be highly amenable to treatment. And could you foresee that if you go for genomics, that you would also do metabolomics on the side? And with that, I mean, you would have an immediate readout of what the genetic variant is actually, what kind of effect or no effect it's having on. And you could also do this in blood and urine. Do you think this might be helpful or too expensive?
Yes, I would. It's always very important that if you have one method, you want to confirm it with another method. So indeed, it would be ideal if you could also use something with metabolomics to see if it's actually, what is actually the result of it. Because now we indeed identified in chapter 3, we have those variants. They are sometimes pathogenic or likely pathogenic, but we still have to figure out what it actually means for the disease. And that's the next part of the translational work, which still needs to be done. And if you would then confirm it with, like, for example, blood analysis or urine analysis, which is, you know, not as invasive as CSF or scans, ultrasounds, and all things combined, you would actually already make the whole process less invasive for the patient. So I would say that's a really good step in the way forward.
Thank you very much. I appreciate how much you think from the patient perspective of decreasing burden. I enjoyed the discussion, and I would like to give the word back to the prorector.
Thank you. The opposition will now be continued by Professor Dr. Koen Poesen. Also, thank you for making the long trip from Belgium, Leuven. Having a chair in lab diagnostics for neurological and immunological diseases at the University of Leuven.
Thank you, Mr. Prorector. It's a pleasure to be here in Maastricht, and it was also my pleasure to read your thesis and to witness your presentation today. So I have a few questions for you, dear candidate. The first one relates to your chapter where you investigate the prevalence of surface neuronal autoantibodies in patients, your 45 patients who were diagnosed with psychosis or who experienced an episode of psychosis about 2.5 years ago. I wondered, in the case of an autoimmune nature, would one not expect that the disease would not be confined to only psychosis, but also in the period of 2 years, on average follow-up, would have developed other symptoms related to the autoimmune nature of the disease, given the subacute nature of an autoimmune disease? So can you comment on that?
Dear highly esteemed opponent, thank you for your kind words and your question. You're totally correct. So, not sure when, but at least 10 years ago, 12 years ago maybe, autoimmune encephalitis was indeed first discovered. And then the idea was that it was mainly present in young females, and they had a really specific onset of the disease with more feverish symptoms. Then they would develop psychosis, and then more neurological symptoms such as epileptic seizures, and then sometimes even coma and death. However, we did notice, and during all those years, that actually it's not so simple. So there are patients who have more symptoms at onset. So the fever, also headache is one red flag, which we call also in general discussion that clinicians should pay attention to. But there are also patients actually described within literature that have more chronic disease. So they have a more mild form of the disease in the sense they do not develop coma or death. And they have, for example, an episode, because the antibodies also have a half-life value. So it's even possible to have like several episodes of those autoantibodies and then the symptoms that are the result of that. So it's very important that, for example, a clinician not only asks about the current symptoms that these patients experience, but also more into the prodromal symptoms that these patients may experience, because that might actually give a hint of indeed such an underlying disorder. So for example, the inborn errors metabolism, you often see that these patients already developed intellectual disability during childhood, but it was missed. And then looking back later or earlier in life, you actually do see that these symptoms are present. So I would say that it's not as simple that we do have a really specific development of symptoms, although in that case, those are probably earlier detected.
Okay, thank you. I have another question, more technically related towards your immunohistochemical analysis on red brain, where I noticed that you diluted your serum 1 and 200, right? And your CSF 1 and 2. So 1 and 200 is not really, at least in clinical routine practice, standard dilution to start with. Okay, so how did you validate this starting dilution? And would you expect this starting titer to maybe more specific towards your findings? Or on the other hand, other side of the coin, would you miss also some cases by using this kind of starting titer?
That's a very good question. That's indeed, what we usually do is we start with a specific titer, which is actually established already before I started my PhD. So there's a lot of hard work for years to actually establish this protocol, which we now like kind of standardized and now use in practice. But actually, if a patient is positive, or if we suspect that these autoantibodies are present, we do a dilution series. So in that case, actually, so we not only do the cell-based assays in the neurons, but first, what I usually did is indeed the dilution series to see what's going on, and also to see if we can see an increase when we have a less diluted samples. So that's one of the, which is not written down actually, but what we do in practice to make sure that what we see is actually there. But I can tell you that if a patient is positive in the CSF, which we actually seen during the summer, you can actually see it by eye, if you're lucky, that the staining is positive. But of course, not every patient is that positive, because, which I also wrote in the discussion of chapter 4, some patients may actually manifest with autoantibodies in their blood and CSF, or CSF only. So
yeah, that relates to my third question. So it's a good introduction to this, where you referred, especially in your discussion, to reference 42. It's a paper of Anthony David and Blackman in Journal of Neurology. Can you comment on that paper? What kind of paper was it, and are there flaws in that paper, or what are the strengths? Where you based your conclusion on that low or serum-only positive samples for NMDA receptor are more likely to develop a psychotic phenotype, whereas with high titers also develop those other symptoms. So can you be somehow critical about this paper, or what the strengths were, and what the nature of this paper was? Can you comment?
Which paper specifically?
Reference 42, it's a paper you discussed in your chapter on your findings on the NMDA receptor antibodies,
Yes.
where you discuss the fact that when you can retrieve them only in serum, it has also some kind of relevance, which is actually a topic of debate, right? It's
Yes.
not that clear. But you referred to that paper, but can you comment on that paper? What kind of paper it was and what are the strengths, the flaws of that paper? Because it's a very controversial issue that only serum NMDA receptor antibodies are clinically relevant or not.
Yeah, so indeed, I agree with you that debate is going on for years, whether or not antibodies that are low positives in CSF or not even possible in CSF actually are pathogenic. This study, but it was not actually the only study who showed that. To be honest, I do not know all the details of the studies anymore.
No, it was a meta-analysis, right?
Yeah,
So
yeah.
it looked at different studies, and based on the different studies, it extracted those findings about those cases where it only antibodies in serum, right? But I think the flaw of that study was that they did not mention any titer, etc., and the clinical analysis maybe of some papers they refer to are also not that excellent. So how would you design such studies to pinpoint if serum-only antibodies are clinically relevant? How should you design such a study?
So first,
In psychosis, right?
So first, that's actually quite a challenge because first you need, of course, enough positive patients in order to extract those antibodies from their blood or CSF. And ideally, you always want to do an animal study in which you then eject those autoantibodies and see what happens to which usually rats or mice. However, it's really difficult to measure psychosis in a rat or a mice.
But
or a
from a
mouse.
clinical point of view,
Yeah.
would you design a clinical study? So you have patients with psychosis and NMDA receptor antibodies present in blood, and what kind of course would you include as well?
So again, in this case, you would need a really, really large cohort because then you would need patients that do have these autoantibodies in blood and CSF. So that would mean probably thousands of patients that you need to screen.
Yeah. And ideally also without any antibodies, right?
Yes, and
To
of course
see whether-
controls, let's not
yes,
forget those,
yes,
which is
okay.
very important.
Yeah.
And then you need a standardized scales which needs to be used so you can actually compare the patients with their symptoms. You would need neurological examinations to detect also the subtle neurological
And would
symptoms.
response to therapy be important in this case, you think?
Well, if you want to do it perfectly, you could use the screener in chapter 2, and then first, maybe that would actually be beneficial to limit the number of patients needed. That you first screen, okay, these patients, for example, have visual hallucinations or other atypical symptoms, and then in the end, if you think, well, these are clinically suspected of autoimmune encephalitis, NMDA receptor or otherwise, and then do the analysis, the immunohistochemistry, cell-based assay, neurons, with all the other stuff, and then in the end you would have a perfectly designed study in which you may be able to draw conclusions.
Let's hope that someone will do
Yes,
this kind of
and
thing.
get
Thank
funding
you.
for that.
Thank you. Now the opposition will be continued by Dr. Friederike Erhardt from Maastricht University and having a role in bioinformatic analysis in rare genetic disorders.
Thank you, Mr. Prorector. Dear candidate, first of all, let me also congratulate you. It was a highly interesting thesis to read. I was especially intrigued as a bioinformatician and data scientist by the amounts of data you generated. First, are you familiar with the term fair data?
Dear highly esteemed opponent, thank you for your kind words and questions. A bit, but could you tell me more a bit?
It's about making data findable, interoperable,
yeah.
accessible, reusable, so basically preserving data, annotated properly, and storing it properly so that it could be possibly reused. So let me ask you, what steps did you take so far to make your data fair, or do you plan to do?
Too little, I would say, if you look at the guidelines of the university. But I know that, for example, we are able to share the panel that we used for chapter 3, so that the other research teams could use the same panel, or actually they would like to have to use the improved version, of course. I know that also with clinical data it's always really difficult because, of course, you have anonymous data, but you also have pseudonymous, and that makes it really challenging because if you, of course, in these studies we do not have the names of the patient, but we give them a certain code. But if you would combine all the different information that we have, you might be able to still trace back who the patient actually is, and that's still really a challenging factor in making data fair. And I think even at the university, they're still figuring out how to deal with that. But I do know there are platforms for that. But that will be something I can look into now after the defense.
I'm looking forward to that, because one thing that intrigued me also when reading your thesis is that you basically have a multi-omics thesis divided in single omics chapters. So
Yes.
I would be highly interested to see one day to have your genetic data, your next generation sequencing data results. You have phenotypic data, you have the description of the patients, and you have also some steps in between with antibodies, proteins, not really, but your metabolites, your lipidomic study. Would it be possible, based on your existing data, to combine these in one multi-omic study? Would that make sense?
To be honest, I'm not sure if that would make sense, because indeed also in a general discussion we discussed whether it would be feasible to just do a whole panel for patients with atypical symptoms. So for example, we test for IEMs with the NGS, and we do the analysis for the autoantibodies, but then again, usually these, other than psychosis, which is the common symptom of course among these patients, you do see that, for example, also it costs a lot of money. It may actually not result in less burden on the patient if you're still going to do all those tests, right? So you would like to limit them, but get the most out of it. So I would not think it's feasible to combine all of them, because having an inborn error of metabolism is actually really different from an inflammatory disease. So I would really emphasize that clinicians really need to focus on identifying the symptoms present in the current situation, but also earlier in life, and then based on that make the decision which panel or which research actually makes the most sense.
How would that multi-omics study look like? So which patient cohort would you choose, and which omics data would you choose for that?
I think you're now referring to chapter 6, right? So of course this is still really a novel path, so we still actually need to confirm that the findings, in this case, for example, these lipids, the phospholipid and the sphingolipids that are actually either elevated or reduced in these patients, is actually related to the psychotic symptoms. We do not know that yet, because the next study would actually to determine if we can replicate our findings, can we still find these findings in patients with psychosis, but also do they differ, for example, from patients with Alzheimer's disease or any other cognitive affected diseases, because it might actually be that these changes in lipids are not related to the psychosis part, but by accident related to the cognitive symptoms that some of these patients might experience, or if it's really different from an inflammatory disease such as MS. So you would actually use those changes to see if they're specific for the disease, and we do not know that yet. So definitely further research is necessary in order to establish that. And in that case, you can get a step further and would maybe screen for those in patients with psychosis.
Okay. Let us stay in chapter 6. I'm working a bit more with gene expression studies, and there I observe often that the changes in gene expression between human patients or human individuals are often larger than the changes induced by the disease. Did you observe that with the lipidomics as well?
Sorry, could you repeat that?
The changes between human individuals, between samples, are between the humans often bigger than the changes between the disease and the control group.
So actually we did the PCA plots, which actually probably makes sense to you. So you can actually see there was clustering. So there was a clustering of patients with psychosis, and there was a clustering of controls. But of course it was not perfect. You know, that's usually never in research. But there were some patients that were more going towards the controls, for example. So we were actually quite happy that we saw this effect. But it's not always the case, of course. In this case, for lipids, we also learned a lot of things, but also that it's really important to have those samples, store them in the same manner, measure them on the same date, because these are all influences very important on the measurements. Yes.
Okay, then let us move a bit to the table with the autoantibodies. I read those tables with also great interest. I was wondering, did you check whether any known schizophrenia or psychosis genetic risk genes are among them? among those targets, so that they can be both genetic risk genes, but also be targeted by autoantibodies and cause the disease?
That would be a very good idea for a following up study. We did not do that, but it would be actually, I think it's quite interesting to determine if you have a large cohort and you have enough patients that are positive for the autoantibodies, if you can determine if a subgroup of these patients, for example, was already genetically affected. That would be very interesting to determine, to see indeed if there's a link between the two. But I'm not familiar with a possible link yet.
It would be just interesting because both whether they are targeted by autoantibodies or whether they have a genetic disposition, they both would basically inhibit their function, causing the downstream effects. One last question, as a science...
Very short question and a very short answer. We have two opponents to go. And
Absolutely. Science is a living thing, so I guess your review or so, when you finished your data analysis and data writing or so, that was possibly something like 2 years ago with the current speed. Is there anything in your list of red flags that you would revisit now or change or update?
No, I would stick with the screener that we actually proposed with the visual hallucinations, the strong cognitive decline, and the intellectual disability, the abdominal symptoms. No, I would keep all of them.
Okay, thank you.
Okay, thank you. Now the opposition will be continued by Dr. Jurjen Leix, who's online from Maastricht University and having his research area is psychiatric genetics.
Dear candidate, many thanks for a great thesis. I was wondering what you think, or whether you could maybe elaborate on the choice that you made for the lipids that you examined in chapter 6. I think it's great that you put emphasis on lipids, and especially that you do this in an exploratory way, but can you maybe briefly outline why you picked those specific lipids?
Dear esteemed opponent, thank you for your kind words and question. I actually did a lot of pre-research before selecting those lipids. So with this method that we had with the collaboration, we are able to measure 2,000 different lipid species, which is quite unique compared to what other studies have done before. However, for me, it makes the most sense to actually select the lipids that are previously already described in literature to be either associated with psychosis, immunity, and cognitive symptoms. So, for example, we did not look into the triglycerides because we know they're highly affected by diet, by the insulin resistant and diabetes, for example, which, of course, these patients with psychosis often get antipsychotics, and as a side effect, it's possible that they develop insulin resistance. So, for example, that's why we did not look at the triglycerides, and there were some others. So the panel that we actually selected were the ones that were previously associated with, at least in some papers, with psychotic patients or cognitive symptoms or in that area.
And then a more general question, maybe a bit more statistical. How is the chance finding defined in science?
Sorry, I did not get the question.
How is a chance finding defined in science?
If I understood correctly, you're referring to the fact that the chance of finding a possible change in those lipids, how big that chance is?
So just a general question, how would you answer this? So in science, we apply statistics to try and see if we can come up with an answer to a research question. And there's always the risk of chance findings, right? So how do you look upon this risk or caveat of chance findings within the context of your chapter?
Of course, usually what you do, for example, if you do an ANOVA, you do the multiple comparison correction, for example, with Blum-Ferroni, or there any other correction that is applied to. I do believe it's important to correct for that, because indeed it's possible that your findings are just a coincidence, and with such a correction, you limit your the chance of such possible coincidences. However, because it's such a new field, there's not a lot known of what the best approach is to which statistic tests are used. and which correction to apply. So that's why we didn't do it yet. But in a very interesting future study, we're planning to do that. We actually want to combine a more metabolic approach and then ANOVA. So it's a whole new analysis, which actually thought of more the golden standard nowadays, but that was done after, or thought of after we wrote this chapter. So ideally you would do that and then indeed do a multiple comparison, which is strict but not too strict, because that's also a discussion always within statistics. So ideally other people would also research that, so we know we have a standardized protocols for which statistic test to use, which correction to apply, and then actually we're able to get the best results out of that and the best conclusions basically.
Great. And final question, so if you would put your money on some kind of lipid implicated in psychosis, which lipid would it be? And can you also maybe hypothesize why that could be the case, how that specific lipid could have the kind of downstream effects that would lead to psychosis?
Yeah, so if you look at the ROC curves on page 166, I would say the top 5 of that would be very interesting. So among them are the sphingomyelins, for example, and some PE species. However, there was done research that actually looked at the combination of the sphingomyelin species and the phosphatidylcholine species. And I would say if you would be able to correct for things like diet and medication in a really big study, I would be really interesting to see what happens to the sphingomyelin, because it's known that sphingomyelin is, for example, also related to the grey matter changes in patients with schizophrenia in that case. But it's not unique. It's also seen in bipolar disorder, for example. And also to see what happens to the phosphatidylcholine, because, for example, that is also known to be affected in Alzheimer's disease. So that would be my best bet. But also the PE species are also known to be affected in Alzheimer's disease. So ideally you would like to have a panel, for example, with the ROC curves, with high area under the curve, that is specific for psychosis and not visible, for example, in Alzheimer's disease. So you can actually use it, hopefully in the future, to select those patients out and based on those findings.
Okay, thank you. I would like to give the floor for the last member of the opposition, which is Dr. Marina Manet-Damas, with research area autoimmunity in the nervous system of Maastricht University. So the floor is yours for the last few minutes.
Yeah. Thank you, Mr. Prorector. Dear candidate, dear Nikita, first of all, I would like to congratulate you very fast for all your work and your nice presentation. And then I will have 1 or 2 questions. Just so you found that the prevalence of these autoantibodies, even the antigen is not identified, is very low. So which type of steps would you take to try to reduce the likelihood of a false positive?
Dear esteemed opponent, thank you for your kind words and your questions. My idea to reduce the likelihood of a false positive would be to combine the different techniques, what we try to do in chapter 4, so both the immunohistochemistry, the cell-based assay, and the neurons, to make sure that what we see in the immunohistochemistry is actually confirmed by other techniques, which is very important in research. And ideally, you would like to do more than one analysis to are certain that what you find is actually there. So that would be one thing. Another likelihood to reduce false positive would indeed be the help of all the clinicians to do the screening already for us. So actually, we have a lot of collaboration that they do the screening for us and we get the samples, and then we can actually be the most efficient. So we only screen the patient who should be screened, so the ones with the red flags, which we described in the general discussion, and then do the test that actually makes sense in that case.
Okay, thank you. And then once you have identified these antibodies, what will be the relevance of identifying the specific target in terms of treatment? Would that change the decision of a physician to apply treatment A, B, or C?
That's a good question. To be honest, I'm not sure, because of course what you usually do if a patient is diagnosed with autoimmune encephalitis, you give immunomodulatory treatment, which is similar usually. However, I do know that for the diagnosing itself, you need the specific target. So I guess that's more like a set rule rather than actually affecting the treatment, because the treatment for most of them is actually similar. Although I can imagine that if a patient has more motor symptoms, you might actually do something additional to the immunomodulatory treatment. But as I'm not a clinician, they have to figure that out.
Okay, and one last question. Yeah, okay. And then once you have identified these antibodies, how would you prove causality?
So ideally, you would do an animal study and inject these auto antibodies into the rat or a mouse. Ideally, rats, I would say, because they're more easily handleable. And then see what happens. But for psychosis, that's really difficult. For example, depression and anxiety, we do know that there are specific behavioral tests that can be done to see what happened to those rats, and you compare them with controls. But for psychosis, that's really difficult. So if anyone has a great idea to measure psychosis in a rat, I would be nice.
You may finish your answer.
So if we could measure psychosis in a rat, that would be the first step actually to see whether the autoantibodies are causally related to the disease.
Thank you.
Nikita van der Burgt, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return in this room.
Please all rise.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home, it's just questions that you've got. Let's go the extra mile and that. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Traveling so far from home, gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future than we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 7 the west side. No place like home. These questions that you've got, go the extra mile and back. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
gewezen.
Dadelijk komt er zo'n mooie ijzeren, zo'n grijze ding naar binnen, dan weet je dat je het zegt.
Ja, en mag drukker van zijn.
Dan moet je wel.
Ja, dat zou het zijn. Oh, dankjewel. Mijn naam staat erop. Oh nee, niet. Mijn naam staat erop. Ja, ja. Dat zou het zijn. Zie je nog van? Ja. Ja. Nee, als hij op het scherm is, moest hij terug. Ja, man. Oh, we hebben... Koffie is niet echt inclusief, geloof ik. De receptie. Allemaal wijn. Ja. Dat weet ik niet. Nou, jij hebt het al gedaan. Ik weet niet, ik heb eigenlijk nooit opgelet wat ze dan doen. Moeten we het even vragen, wie heeft wel een PhD? Laat ik gewoon staan. Ja. Yay.
We gaan nu door in het Nederlands.
Oké.
Nikita van den Burgt, de commissie hier aanwezig, heeft zich beraden over de kwaliteit van uw proefschrift en de wijze waarop u dat proefschrift heeft verdedigd. En op grond van haar positieve oordeel en gelet op de examens die u vroeger heeft afgelegd, heeft zij besloten u het gevraagde doctoraat te verlenen. De hoogleraar, professor van Amersfoort, is gemachtigd u op de gebruikelijke wijze met deze waardigheid te bekleden. En het woord is aan uw promotor.
Belooft u dat u altijd volgens de beginselen van wetenschappelijke integriteit te werk zult gaan? Eerlijk en zorgvuldig, transparant, onafhankelijk en verantwoordelijk?
Dat beloof ik.
Krachtens de bevoegdheid ons door de wet toegekend, volgens het besluit van de commissie hier tegenwoordig, verklaar ik hierbij u, Nikita Alana van den Burgt, tot dokter te bevorderen en u alle rechten te verlenen, die daaraan volgens wet en gewoonte zijn verbonden. Ten bewijze hiervan overhandig ik u nu de bul, door rector, secretaris en overige leden van de promotiecommissie ondertekend en met het grootzegel van de universiteit bevestigd.
Geachte dokter van den Burgt, beste Nikita. Van harte gefeliciteerd met het succesvol verdedigen van je proefschrift. En felicitaties ook aan de familie. Je hebt ervoor gekozen om je proefschrift The Many Faces of Psychosis te noemen. En dat er vlinders op de omslag staan is geen toeval. Vlinders komen namelijk voor in alle kleuren en ze weerspiegelen ook jouw persoonlijkheid, heb je me uitgelegd. Hoe gelijk je toch had. In de afgelopen jaren heb je met veel passie onderzoek gedaan. Je hebt in het lab gestaan, verschillende technieken geleerd en toegepast en vele uren achter een microscoop of computer gezeten. Ik herinner mij nog goed aan berichtjes met foto's van kleuringen die eindelijk gelukt waren en hoe blij je toen was. In de loop van de tijd heb je door je werk in de 2 divisies en de klinische aspecten van je studies met heel veel mensen samengewerkt. En het was natuurlijk te verwachten dat dit heel veel communicatie en organisatie van je eist. Maar je hebt ook ervaren dat onderzoek doen betekent dat niet alles verloopt zoals verwacht en onverwachte uitdagingen zich voordoen. Soms misschien zelfs vaker dan je zelf had gewild. Soms werkte je computer niet mee en vroeg heel veel geduld van je. Of was er plots een wereldwijde pandemie. Tijdens de coronapandemie werkten we vaak vanuit thuis, waardoor onze besprekingen helaas online moesten plaatsvinden. Je zat vaak voor de computer met zo'n heel grote kleurrijke mok. Maar wat nog vaker in beeld kwam, was iets anders. Je katten. Bijna bij elk gesprek liep plots een kat over je toetsenbord. En zonder van de camera weg te kijken, pakte ik de kat en zette hem opzij. Maar meestal was dit slechts een tijdelijke oplossing, want kort daarna liep weer een kat door de camera. Soms vroegen ze ook je aandacht door aan je been te krabben. Weet je, eigenlijk weet ik nu alsnog niet hoeveel katten je hebt. 3. Ja, het leek alsof je 10 had, maar inderdaad, ik dacht daar ook, misschien zijn het maar 2 of 3 die gewoon heel graag je aandacht vroegen. Dus tijdens COVID kwam ik erachter wat voor een dierliefhebber je bent. Hè? Katten, konijnen en je hebt zelfs een alpaca geknuffeld. Stap voor stap heb je aan je studies gewerkt, vanuit thuis en wanneer toegestaan ook in het lab. Maar ongeacht waar je was, je stond altijd klaar voor je collega's, wat iedereen heel erg waardeerde. In het laatste jaar liet je ons nog een nieuw facet van je persoonlijkheid zien. Terwijl de meeste studenten stress ervaren bij het afronden van het proefschrift, werd jij er juist rustig van. Oh, dat gaat wel lukken, zei je tegen mij. En deze rust kon je vasthouden tot 3 weken voor het indienen van je proefschrift. Toen was het vrijdagavond, bijna 5:30. Ja, weet je het nog? Ja. Ik kreeg een bericht van jou met de vraag of we konden bellen, omdat je de analyse opnieuw gedraaid had en helemaal in paniek was. Dacht ik zal ze maar snel bellen om te horen wat er aan de hand is. Nikita, als je collega's gevraagd worden om jou met 1 woord te beschrijven, dan hoor je vaak hetzelfde antwoord. Wervelwind. Je bent inderdaad een kleine wervelwind. Als jij een ruimte binnenkomt, weet iedereen dat jij er bent. Je vult meteen de hele ruimte met je lach. Maar als wervelwind heb je nog een andere eigenschap. Je kunt heel snel praten. Je kunt heel veel in een heel korte tijd vertellen. En dat al wanneer er niks aan de hand is. Dus wat moest ik dan die vrijdagavond verwachten van een Nikita die in paniek was? Dus ik zal het gesprek in een trager tempo samenvatten, zodat het te volgen is. Dus je had de analyses opnieuw gedraaid en je p-waarde was veranderd. Ja, oké, dat kan gebeuren. Ik vroeg naar de nieuwe p-waarde. Je wist de p-waarde niet precies uit je hoofd, maar die was in ieder geval niet significant. Ik vroeg of je de oude p-waarde nog wist. Nee, dat wist je niet, maar die was niet significant. Er viel even een stilte, want ik moest even nadenken wat jij als probleem zag, want ik zag het op dat moment niet. Ik zei, dus de p-waarde verandert van een niet-significante waarde naar een andere niet-significante waarde. Ja, een cijfer naar de komma is anders, zei je. En weer viel een stilte. En toen begreep ik dat er geen probleem was. Tenminste niet met je data of analyse. Want het enige probleem was dat jij op dat moment niet zag dat er geen probleem was. Dus ik vroeg aan jou, verandert dit iets aan de interpretatie van je data of je conclusie? Nee, eigenlijk niet. En weer een stilte. Zodat je even kon verwerken wat je zelf net gezegd had. Maar het kwartje viel nog niet. Mijn volgende vraag was of je veel moest herschrijven of aanpassen. Nee, eigenlijk niet, enkel de p-waarde. Oh, en toen begon je te lachen. Ik kon echt aan je stem horen hoe de hele stress van je afviel. Je hebt dus zowel de leuke kanten als de uitdagende kanten van onderzoek doen leren kennen. Naast je onderzoekswerk heb je ervoor gekozen om PhD-vertegenwoordiger te worden en je hebt veel tijd besteed aan lesgeven. En al snel bleek dat onderwijs geven ook een passie van je was. Met veel geduld en enthousiasme begeleidde je individuele studenten tijdens stages, maar ook grotere groepen. Je haalde hier veel energie uit en ook motivatie. Zelfs zoveel energie dat je steeds meer een mogelijke toekomst in onderwijs zag. Nikita. In de loop van de afgelopen jaren mocht ik de vele facetten van jouw persoonlijkheid leren kennen. En sommige eigenschappen zijn bijzonder sterk gegroeid in deze tijd. Je kunt eigenlijk zeggen dat je toen zoals kleine onervaren rups voor onze deur stond. Een rups die het leven van patiënten wou veranderen. En om dit te bereiken heb je onderzoekdoel gekozen en je hebt hard gewerkt. In juni 2021 plaatste je een bericht op een social media platform.
Oh god.
En over iets meer dan een jaar hopelijk dokter voor mijn naam. Ik denk dat ik het zelf niet kan geloven totdat het daadwerkelijk zo is. Het duurde iets langer, maar kijk waar je nu staat. Je staat vandaag hier op de Minderbroedersberg als dokter. Als uitgegroeide, stralende vlinder. En je kunt trots op jezelf zijn. Je bent klaar om naar nieuwe avonturen te vliegen. En je eerste reis gaat naar de psychologiefaculteit, waar je je passie voor onderwijsgeven gaat volgen. Ik wens je heel veel succes en plezier op deze en alle toekomstige reizen. En als er eens een wolk of een storm opkomt die het vliegen en je reis moeilijk kan maken, dan denk eraan dat achter de wolken altijd de zon schijnt. Proficiat.
Dankjewel.
Ja, dokter Van de Burgt, namens de Universiteit Maastricht feliciteren we u met de behaalde dokterstitel. Ik was onder de indruk van de defense, van de wetenschappelijke interactie. Ik heb ook met interesse naar het boekje gekeken. Het was helemaal in het Engels, behalve 1 zin. Pagina 5. Walt Disney. Ja, dat is toch een Amerikaan, dus ik dacht eigenlijk moet dat ook in het Engels zijn geweest, maar dat heb je vertaald. Al onze dromen kunnen uitkomen als we de moed hebben om ze na te streven. Nou, ik denk dat vandaag een droom is uitgekomen en daarmee gefeliciteerd. En een tweede, dat was ik ook zelf, omdat ik zelf veel heb hard gelopen in het verleden, dat was dan een stelling 9 van Usain Bolt. Ja, die liep wel echt wat harder dan ik dat kon. The sky has no limits and neither should you. En Usain Bolt die deed altijd zo.
Ja.
En nou weet ik waarom. Hij wijst naar die sky en dat is zijn stelling. Dus dat heb ik ook alweer geleerd vandaag. Dank je wel daarvoor. Nog een paar logistieke zaken. Omdat we iemand online hebben, gaan wij zo dadelijk even hier ons verzamelen voor een foto, zodat op de achtergrond de opponent, Dr. Luijks, ook in beeld komt. Hij zal aansluitend ook de gelegenheid krijgen om u online te feliciteren en dan kunt u daar antwoorden. U kunt dan naar de receptieruimte gaan. Wij hebben dan nog 1 stap te gaan, want we gaan daarna met het hele oppositie- en promotieteam en de nieuwe dokter naar de trappen hiervoor. En dan gaan we een foto maken en daarna komen wij naar de receptieruimte. Hiermee wil ik deze vergadering afsluiten. En gaan wij nu over naar het foto's maken.